We've all heard those words, right? Tumor, growth, cancer. They get thrown around a lot, and let's be honest, they can be confusing and, well, a little scary. But what if we could cut through the confusion and really understand what separates a harmless growth from a dangerous one? Well, that's exactly what we are going to do.
So, let's just dive right in with the big question on everyone's mind. We hear these terms used almost interchangeably, but are they really the same thing? Is every single tumor cancer? Is every growth a tumor? It's time to finally clear this up. To really get to the bottom of this, we have to start at the very beginning with the foundational term that connects all of these concepts: neoplasm. This is the scientific root of the entire conversation.
Neoplasia, it literally just means new growth. But, and this is the most important part, it's not normal growth. This is growth that's gone rogue. It's abnormal, it's excessive, and crucially, it just doesn't stop, even when whatever started it is long gone. You can think of it like a car with the accelerator stuck to the floor. The growth just keeps going and going, completely ignoring the body's normal stop signals. This is the starting point for any tumor.
Okay, so once this new growth gets going, it hits a fork in the road. And as you can see here, it's going to follow one of two very, very different paths. And those two paths have vastly different implications for a person's health. And this brings us right to the core of it all, the big distinction between benign and malignant. On one side, you've got benign tumors, and on the other, you've got malignant ones. They might start in the same place, but believe me, their stories end very differently.
All right, here's a simple way to think about it. A benign tumor is like a quiet, well-behaved neighbor. It might get bigger, sure, but it stays in its own yard. It stays localized. A malignant tumor, on the other hand, and this is what we call cancer, is the bad neighbor. It has no respect for fences, it can invade and destroy the surrounding property, and it can even send out seeds to start new colonies all over the body. That process of spreading, that's called metastasis.
And you can literally see this difference under a microscope. Take a look at the image on the left. That's a benign tumor. See how orderly and predictable it is? The cells look pretty similar, well organized. Now look at the right. That's a malignant tumor. It's just cellular chaos. The cells are all jumbled up, different sizes, different shapes. This visual contrast is actually the first major clue that pathologists look for. So, how do doctors actually tell the difference between the orderly neighbor and the chaotic one? Well, pathologists have a sort of checklist. They look for 4 specific telltale signs that, together, build the full picture. These 4 characteristics are the absolute cornerstones of diagnosis. We've got differentiation, rate of growth, local invasion, and metastasis. Let's walk through each one so you can see exactly what they mean.
First up is differentiation. This is all about identity. Do the cells still look like where they came from? Cells in a benign tumor are what we call well-differentiated, which is just a fancy way of saying they still look a lot like the healthy cells they grew from. Malignant tumor cells, though, they start to lose that identity. They become poorly differentiated, or even anaplastic, which means they look so primitive and disorganized they're barely recognizable, just like that chaotic image we just saw. Next, let's talk about rate of growth and local invasion. Benign tumors usually grow pretty slowly and, really importantly, are often contained within a fibrous capsule, like a little biological wall that keeps them from spreading. Malignant tumors, however, can grow rapidly. They don't have that capsule, and they actively invade the tissue around them. There's our bad neighbor again, breaking through the fences.
And finally, metastasis. Now this is the big one. This is the ability of tumor cells to break away, travel through the bloodstream or the lymphatic system, and set up new tumors somewhere else in the body. The presence of metastasis is the absolute, unequivocal, #1 sign of a malignant tumor. It's the defining feature of cancer. It means the cells have lost their normal stickiness and learned to survive on their own. That ability to spread is what makes cancer so incredibly dangerous. Okay, now that we can tell them apart, let's look at how they're named. Because in medicine, a tumor's name is basically a code that carries vital information about its nature. This quote says it all, really. The name of a tumor is like a compact diagnosis. It can tell a doctor where the tumor came from, whether it's fat or a gland or fibrous tissue, and whether it's likely to be benign or malignant. So let's look at a simple but really powerful naming rule. Generally speaking, if a tumor's name ends in oma, it's benign. A lipoma is a benign tumor of fat tissue. A fibroma is a benign tumor of fibrous tissue. Simple enough, right? But if you see sarcoma or carcinoma, that signals malignancy. So a liposarcoma is a cancerous tumor of fat, and an adenocarcinoma is a cancerous tumor of glandular tissue. The name itself is the very first clue.
All right, we've covered the definitions, the differences, even the naming system. But why does this distinction matter so much? Because understanding it is absolutely central to tackling one of the biggest health challenges in the entire world. Take a look at this number. It's staggering. According to data from 2018, cancer, or malignant neoplasia, was responsible for 1 in every 6 deaths globally. This isn't just abstract medical terminology. It is a reality that affects millions and millions of lives every single year. And this data breaks down cancer's impact even further, and it shows why knowing the origin, remember our naming system, is so critical. This 2019 data highlights that for men, the highest incidence is prostate cancer. For women, it's breast cancer at a striking 30% of new cases. So you see, distinguishing between different types of neoplasms isn't just a lab exercise. It's essential for prevention, for diagnosis, and for finding the right treatment. So, we've learned how to define a neoplasm, we know how to distinguish the benign from the malignant using those 4 key hallmarks, and we've seen why this all matters on a global scale. We can now identify cancer's many forms with incredible precision. But this just leads to the next, even deeper question, doesn't it? What is it that actually flips the switch inside a perfectly normal cell, causing it to ignore all the rules and begin that journey of uncontrolled new growth in the first place? Well, that's a story for another time.